All right, today we're going to cover chapter 16, which is regulated transcription in prokaryotes. So by the end of this chapter, you should be able to summarize the mechanisms by which transcription is regulated, distinguish between positive and negative control, explain the details of the lac operon regulation, describe the role that RNA plays in the control of gene expression, and interpret experiments aimed at understanding gene regulation. So this chapter, we talked about transcription already, and so what's different about this chapter? So now we're talking about regulated transcription. And so the idea between regulated transcription is that there are certain environments, let's say, that are different from each other. And so in environment 1 and environment 2, you have expression of a gene. So you express a ton of mRNA for a gene, whereas in environment 2, you don't express the gene. And so the idea is that you can turn on and turn off transcription as needed. And so this is a very important general principle for understanding multicellular organisms. So humans, for example, have a large number of different cell types. So we have different tissues. We have heart tissue, we have skin tissue, we have brain tissue, and all of those tissues and all of those cells are very different from each other. And they're different even though they have the exact same amount of DNA and the exact same DNA content. So why is it possible that you could have different cells? And that is simply because it's possible to turn on and turn off expressions of genes. And this is also true of bacteria as well. So even though bacteria is a single cell, it's able to turn on and off expression of single genes. So it has 4,000 genes. A typical bacteria has 4,000 genes, and it's able to turn them on and off as needed. So an example of this is shown on the right. So this is how a cell is able to turn on and off expression of genes needed for the cell cycle. So we're looking at a bacteria called Caulobacter crescentus, and it is studied for its cell cycle. So it starts out life as a swarmer cell. It differentiates into this stalked cell type, and then it undergoes cell division. So you can see it becoming two different cells. So it's replicating its DNA, it's growing, and then eventually it grows and segregates its DNA and splits into two cells. So if we look at how genes are expressed during the cell cycle, we see that genes have a very particular time that they're turned on and turned off. So yellow indicates when the genes are turned on. So this is how much mRNA is actually within the cell. Blue indicates when there's not any mRNA within the cell. And so you can see genes are turned on and off as needed. So for example, when the cells are building a flagellum, there's a suite of genes that are turned on in order to build that flagellum. When the bacteria are building the stalked organelle, there's a suite of genes that are turned on to help it build that stalk. So this is possible because the bacteria have evolved mechanisms by which to control gene expression. And that's what we're going to go with in detail in this chapter. So the most common way to regulate gene expression in bacteria is by influencing the initiation of transcription. So whether or not that RNA polymerase is able to bind to a promoter and start to initiate transcription. So we talked about breaking down transcription into initiation, elongation, and termination. So typically what's regulated is whether or not initiation occurs for a gene. So the rate of DNA synthesis can be increased or decreased. So you can have factors that come in and increase expression. You can have factors that come in and decrease expression. So transcriptional regulation involves the action of two main types of regulatory proteins. So it's important to remember that the mechanism that we're talking about are proteins actually coming in and binding to DNA and binding to RNA polymerase. So one of these factors are called repressors. So these are molecules that bind to the DNA and inhibit transcription. They turn down transcription. Activators, these are proteins that come in and bind the DNA to increase transcription. So again, you can turn on and turn off transcription. Activators are the ones that increase. Repressors are the ones that decrease. Negative control refers to transcriptional regulation by repressor proteins. So we're going to see this in some of the examples that we go over. And then positive control refers to regulation by activator proteins. And so, when we're talking about transcription, we're talking about transcription of a single gene. And just as a reminder, the upstream region of the transcriptional start site is the promoter. So this is where the RNA polymerase can bind. And then we can also have sequences where DNA factors, additional transcriptional regulators can bind. So here at the activator binding site, we see we have an activator binding to this region, and then it's able to turn on transcription. So if this activator is not present, transcription does not occur. So it does not recruit the RNA polymerase to have transcription. So this is an example of positive regulation. You have an activator come in to increase transcription. The opposite is negative regulation. And so this is when you just normally have transcription, but you turn it off by having the repressor. So in this case, the repressor binds to this operator sequence. So this is still part of the DNA sequence. There'd be a run of nucleotides that is recognized by this repressor, and by this repressor binding to this region, it's able to prevent the RNA polymerase from coming in and binding to this promoter and turning on transcription. If this repressor is not bound to the operator, then the RNA polymerase is able to come into the promoter, and now we have transcription turned on. So this is an example of negative regulation. So this chapter, the details of this chapter, is we're just going to go over 3 different examples of how a gene is regulated. So we're going to start out with the lac system. So this is a set of genes that's necessary for metabolizing lactose as a sugar source. We're going to talk about the cap system. So this also regulates the lactose genes, but it does it in a unique way. And then we're going to talk about the tryptophan system. So this is genes that are required for creating tryptophan, for the biosynthesis of tryptophan, which is an amino acid.